Hello, everyone. Welcome to Neuroanatomy, Metabolism, and Neurotransmission in the human and primate brain session. My name is Hiromasa Takemura, who is going to chair this session together with
Paul Toussaint.
And we have some very small number of instruction. So each speaker have 10 minutes. Then after that, we have 2 minutes for question discussion. Then this is not very big room. If you have a question, we encourage you to go to the microphone. But each person, please just restrict your just number of questions to just one question, please. Then you also have an option to post question in the app. Okay, so let's get started.
Okay, so our first speaker for the session will be Rebecca Glarin from University Melbourne, who will speak to us about the ultra-high field functional brainstem imaging of nuclei responsible for blood pressure control.
Thank you. So today I'm going to be presenting a bit about blood pressure control, and excuse me, but my blood pressure is quite high at the moment, so if only I could actually control it. So a bit about sympathetic drive. It's a key component of the baroreceptor reflex, and this reflex controls blood pressure within the body. This happens through input from the baroreceptors, which are the arterial stretch receptors found in the carotid sinus and the aortic arch, and they help to detect the beat-to-beat blood pressure changes in blood pressure. And they signal to key brainstem circulatory to alter the sympathetic and parasympathetic drive to the heart and the blood vessels. So there's been decades of preclinical work looking at this circulatory, and this has identified the signals travelling from the baroreceptors up the glossopharyngeal and vagus nerves to the NTS, onto CVLM, RVLM, and down the intermediolateral cell column. So whilst this has been fantastic work, it is all preclinical, and what has actually happened with this is it's all been through electrical or chemical stimulation directly into the brainstems of these animals. So this is not translatable to human research. But when we do look at how this has been investigated in human research, they are through evoked responses. So kind of a little history of the few studies that have done this. So there's a Valsalva study on the 1.5 Tesla back in 2002, and this increases the sympathetic nerve activity and therefore the blood pressure with this Valsalva technique. And this showed extensive regions in the cerebrum, brainstem, and cerebellum. Then we moved into 3 Tesla as imaging progressed, and Gerlach did this study with chemical stimulation to increase blood pressure, so an infusion of a drug. And this showed this really dramatic response within the brainstem, which was very exciting, but it also makes you consider, I wonder if we can tease these nuclei out with better specificity. Then we move on to the first 70 study, looking at these responses in the brainstem, and this was an exploration breath hold study, and it really teased out these regions within the PAG that were responsible for controlling this blood pressure. But these, again, are all evoked responses through Valsalva. There's some negative body pressure suits, apnea, things like that. So what if we strip this back to spontaneous activity? What drives your body to control your blood pressure when you're not doing any particular activity, you're just at rest? What is that organic kind of basic science that's driving that process? So we can actually look into this with something called muscle sympathetic nerve activity, and the technique for recording this is microneurography. So muscle sympathetic nerve activity is composed of the burst of action potentials which supply the highly vascularized skeletal muscles throughout the body. And it's tightly coupled with the arterial baroreflex, and by controlling blood flow to the muscles, it allows for vasodilation, which contributes importantly to that beat-to-beat regulation of the blood pressure. And you can see here that as the blood pressure regularly cycles through, it's temporally coupled with these increases in this MSNA, or these bursts that are controlling it. And the way that we can record this is through something called microneurography, which involves the insertion of a tungsten microelectrode into the muscle fascicle within the nerve, and then we amplify this signal, and it comes out as this trace that you can see here. So this technique was then coupled in 2010 with fMRI. So the participants are lying in the scanner at rest, recording their activity and coupling it with their bold response. And this was a really exciting study. It had 5 participants, and it managed to identify increases in RVLM and CPA, while decreases in CVLM and NTS. And this is what we would expect from that feedback loop that we know that occurs within the brainstem. But this was really exciting results. But then as we progress and science progresses, we think, what can we do with this? How can we investigate this further? Can we actually break it down even more? So we considered doing this study at 7 Tesla. And when we looked at that, we could see on this 1.5 Valsalva study, it's not very specific, but you can see that it's RVLM. It matches the location you would expect. Spontaneously at 3T, we get better specificity, and we're marking out that RVLM there, mapping out. And then when we look at 5 participants of this same technique between 3T and 7T, you can already see that you're starting to get these brainstem nuclei with much better specificity. And this was the kind of support that we needed to drive our study forward. So our study design was 20 healthy participants, normotensive, and they lied on the scanner table for 3 hours, having microneurography in their left perineal nerve. We recorded the MSNA and amplified it to 20,000 times, and to ensure uniform levels of attention, all participants were watching the same animated G-rated movie. And then we scanned a structural scan for co-registration, and then a 1 millimetre isotropic brainstem-only slab, which was 4-second TR each acquisition, and we ran it for half an hour. Now, there might be some naturalistic people in the room. I'm not really sure, and I know you're all thinking, what movie did you watch? We didn't consider that, that part of it in our parameter design, but what we did think is we just want people to stay awake, because when you fall asleep, your activity decreases. And we wanted people just to be in a generally happy, relaxed state, not thinking about their thesis, not thinking about why their results didn't work, just, going back to their childhood when things were less stressful. So they all watched Madagascar. Yeah. So then moving into our study design, the considerations for this, so there's a 1-second neural delay from the brainstem to when recorded in the perineal nerve, travelling down the C fibres. And we know there's a 5-second hemodynamic delay from when an event occurs to when it shows up in the bold response of the imaging. And so we know that when we record the nerve signal in the leg, it's 4 seconds until we see that bold activation in the brain. And this was a driver of a model of a 4-second model between our nerve recording and our bold response. So this is what the difficulty of doing some things in MRI, and I mentioned that we amplified this signal by 20,000 times, that unfortunately when the magnet is on, the gradients are working, it completely obliterates the signal. So we've got these lovely bursts, scanner gradients are working, it's gone. Lovely burst again, gradients are working, it's gone. So we record the signal for 4 seconds, acquire a data set for 4 seconds, signal for 4 seconds, etc. So even though they're in the scanner for half an hour, it is a 15-minute data set. And then what we do is we divide the 4-second acquisition into 4 1-second epochs, and then each of these seconds correlates to a region within the brainstem for our analysis model. And you can see another example of that here. So there's a burst in this epoch. It relates to this region of the bold analysis. So as we move into our results, we ended up with 14 healthy subjects. Unfortunately, some people did fall asleep and had low sympathetic nerve activity. A couple of people moved. It's understandable. It's a long time in the scanner. We analysed the data with SPM, with distortion corrected, spatially smooth, aligned to MNI, and we used astute brainstem analysis, which I tediously had to trace the manual brainstems of all these participants. So then we decided to look at this data in a slightly different way from the 3T study. They looked at people with low burst count versus people with high burst count. We actually unpacked it a bit further to kind of strip it back to look at if there was a burst, what was happening in the brainstem in the bold activity. Then we looked at does the amplitude of the burst correlate with the changes in the bold activity? So 2 different analysis models. So firstly, with our burst incidence, we were able to identify all the nuclei that we would probably expect that we know are part of this sympathetic drive process in the brainstem. But probably what was most important to us was that we saw this bilateral RVLM with better specificity than what we'd seen at 3 Tesla. And then when we unpack this further into the amplitude data, we actually managed to find a decrease in NTS and CVLM. The increases in raphe obscurus and dorsal raphe nucleus were also, expected, what we know for the circuitry. But this was really exciting. So what we actually managed to do was tease the data set from the 3T study apart. And it raises the question, are there actually two sites of modulation in the brainstem for the baroreceptor reflex? So Kainbaum hypothesised in 2001, a long time ago, that there was in fact potentially a region in the brainstem that is responsible for how strong the burst is. And then there was another region that was responsible for whether the burst occurs or not, so kind of opening the gate for that regulation of blood pressure. So we actually believe that for the first time in humans, we've managed to use 7T imaging to provide evidence that there are two sites of modulation in the human baroreceptor reflex, amplitude controlled by NTS and CVLM, and burst incidence controlled by RVLM. So our future works with our data set is we also have the same protocol scanned on the whole brain, and we also have the brainstem and the whole brain data sets looking at skin sympathetic nerve activity. So in response to kind of thermoregulation, we also have pain on these participants while recording their microneurography as well. So once we've identified all these key nuclei, we'll then start to map out the connectivity of them in respect to controlling sympathetic drive. I guess you could also say we're going to apply it to some clinical cohorts of like POTS and long COVID, but there's maybe potential to include it in studies into maybe like interoception or people, having difficulty with, that area. So finally, thank you to my group where I work as a radiographer as well as completing my PhD. Thank you to my supervisors. If you'd like to chat more about this, feel free to come to my poster this afternoon. And finally, I just want to say thanks to the brainstems. I think there's about 6 of those 14 brainstems sitting in the audience today. I appreciate the efforts of sharing a perineal nerve and your brainstem with me to help get this data set. So thanks.
Questions from the audience? Yes.
For measuring some of the pressure changes, there's also the just pressure transducers that are pretty noisy. But I'm curious if you've thought about combining those noisier measures to get time, use the electrical measure to get calibrated time that kind of goes through the bold, noisy periods.
Sorry, you're talking about like blood pressure monitors?
Yeah.
Yeah, so that is something that we're looking at bringing into the lab so that we can monitor the blood pressure at the same time. Okay. But we have found that things like the respiration, the pulse, the blood pressure, we can't really use as a regressor in our signal noise because it's temporally coupled with the bold activation. Okay. So we've had to kind of like really strip it back so that we don't regress out any of our signal that we're wanting. Okay. Yeah.
So thank you very much.
Thank you.
Thank you very much.